Hi there, welcome to our revision series on IB biology. We are continuing with 2.2, talking about cell structure, and today we're going to focus on prokaryotic cells. So, for prokaryotic cells, you need to understand the components. You need to understand that the DNA is a naked loop and it has a 70S ribosome. You need to have an idea about the structure of a gram-positive eubacteria, such as Bacillus or Staphylococcus. Okay, so prokaryotic organisms include bacteria and archaea and are small and unicellular. So prokaryotics contain the following: a cell wall, plasma membrane, flagella, pili, ribosomes, the nucleoid, a region with free DNA. So the prokaryotic cell wall maintains the shape of the cell. It also keeps the bacterial cell from rupturing when water pressure is greater inside the cell than outside. And one of the key components of the cell wall that it consists of a carbohydrate called peptidoglycan. Now, the layer outside the cell wall is a capsule, and this is present in some bacteria. This allows it to adhere to things such as teeth, skin, or food. Now, one of the key things is when you're looking under the microscope, gram-positive bacteria have cell walls when exposed to a crystal dye, and these appear violet or blue under a microscope. Gram-negative bacteria do not appear blue or violet under a microscope. So, some bacterial cells contain growths on the outside of the cell wall. These structures are pili, and basically their main function is for attaching to other bacterial cells in preparation for the transfer of DNA from one cell to another, which is utilizing sexual reproduction. So in prokaryotic cells, the cytoplasm occupies the inside of the cell. Now, one of the key components is that there are no specialized areas within prokaryotic cells, so chemical reactions are not isolated from one another. And what this can do is limit the cell's development, and this can be due to interference between different chemical reactions. So ribosomes are present in all prokaryotic cells, and they are the main sites of protein synthesis. So ribosomes consist of two subunits, a protein and ribosomal RNA, and with prokaryotes they consist of 70S ribosomes. So you've got this concept of the nucleoid region. The nucleoid region of a bacterial cell is not compartmentalized and contains a long circular thread of DNA, basically the bacterial chromosome. It's not surrounded by a membrane, and they are all in prokaryotic cells, they are not associated with proteins called histones, and the nucleoid region is basically involved in cell control and reproduction. And then finally, we have this process called binary fission. So basically, prokaryotic cells divide by a simple process called binary fission, and the DNA is copied and results into daughter chromosomes. And then the daughter chromosomes become attached to different regions on the plasma membrane, and the cell divides into identical daughter cells. And a diagram is shown on the right-hand side of the process of binary fission. Thank you very much for listening to this recording on prokaryotic cells in section A2.2. We will be conducting more lectures on our IB biology revision series.